Hello students, now we will start with new topic, that is DNA structure. Structure of DNA was in detail given by Watson and Crick. Structure is given by Watson and Crick. Term DNA is given by Zacharis. The strength of this DNA molecule and the stability of the DNA molecule, it is because of having a special feature called repair mechanism. Why we are discussing this point? Because now we are going to read about the structure of DNA that is not going to change in case of eukaryotes. It is very stable in its structural arrangement. So we are discussing this point. The stable nature of DNA or structure of DNA is because of special feature called repair mechanism. The backbones of DNA structure are pentose sugar and phosphate molecule. The backbones will write. Next, the structure of DNA has two strands running anti-parallel to each other. Running anti-parallel to each other. Here, when they are running anti-parallel to each other, for suppose, one strand is moving in one direction. Since it is a double-stranded coil structure, another strand will be moving in the opposite direction. This term, the direction of movement of the strands, is used with a special term called as polarity. So we can frame out a sentence like, the polarity is from one direction to other direction, that is having some numerical numbers, that is 5' to 3'. I will make two different types of sentences now. Simple form of the sentence is, the direction of DNA is from 5' to 3', and because two strands are there and one is running anti-parallel to the other and other one from 3' to 5' direction. Now, the same sentence we will learn in other way, that is, the polarity of the strands is polarity of strands in double-stranded DNA. In double-stranded DNA is from 5' to 3' and 3' to 5'. Here I have given you two different sentences, but the two sentences meaning is same. Means instead of using the direction of the strands, I am using technically the term polarity. So whenever in a question or in a sentence, if you see the term polarity in case of DNA, just remember that the point is explaining about the direction of the movement of the strands. If we draw the structure of DNA as right-handed helical form of structure, Let's give the names of the folded forms. Suppose think that one strand, here we will talk about the black color shaded one. This is having at one end 5 prime. I will tell you what is that 5 prime and 3 prime now. If the one end is having 5 prime, then other end will be having 3 prime end. In the same way, this strand will be having on the opposite end 3 prime, means 5 prime end is the black strand, black color labeled one. So this end, this will be 5 prime. We already discussed in biomolecules chapter what are names of these small depression like structures. These are called minor grooves. This big depression is called major groove. Again, this small depression is called minor groove. So this is major groove. This one is also major groove. The distance between two major grooves or two minor grooves is considered as one turn of the DNA. It can be either from here to here or from here to here. Between, on one side only we should consider, not from major from here to the major on the other strand. Either between the length between the major grooves on one side, means between two major grooves on one side, or between two minor grooves on other side. That distance is considered as length of DNA strand or one turn. Sorry, length of one turn of the DNA. Length of one turn. Right? What is one turn first? The distance between two major grooves or two minor grooves on one end of the strand is called as one turn of DNA. Now, length of one turn. This length is considered as 34 angstroms. That is 34 angstroms. This 34 number is in terms of angstroms of one turn. Then, what is the number in terms of nanometers? That is 3.4 nanometers. Again, since nano means 10 power 9, so in terms of meters, it is 3.4 into 10 power minus 9 meters. So, number of nitrogen base pairs in one turn. 10 number of base pairs are located in one turn of DNA. So, here we can write 10 base pairs. Base pairs I am writing with small b, small p and s. Coming to the number of nitrogen bases. This one also written as 20 base pairs, sorry, nitrogen bases. 20 nitrogen bases. The diameter when we consider an central axis in this DNA strand, if we go for the value of diameter in DNA, then it is 20 angstroms. Means the distance from here to the distance from here up to here, the one which is straight to this is 20 angstroms. Here I have written base pairs. If we start from one minor groove, here we can write adenine bonding with thymine, guanine bonding with cytosine, again adenine bonding with thymine, cytosine bonding with guanine, again cytosine bonding with guanine. So 1, 2, 3, 4, 5 pairs. Another turn is there. Adenine pairing with thymine, thymine pairing with adenine, adenine pairing with thymine, guanine pairing with cytosine, cytosine pairing with guanine. So we have represented 10 number of base pairs. 1 pair, 2nd pair, 3rd pair, 4th pair, 5th pair, 6th pair, 7th pair, 8th pair, 9th pair, 10th pair. Then I have written here 20 number of nitrogen bases. We will count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20 number of nitrogen bases are present in one turn of DNA. Another important point. Always adenine pairs with thymine. Adenine, here I am writing pairs with thymine. And guanine pairs with cytosine. So again I am using the term pairs with cytosine. But here it is very important to know that adenine always pairs with 2 hydrogen bonds. Pairs with thymine with the help of 2 hydrogen bonds. Guanine always pairs with cytosine with help of 3 hydrogen bonds. Means 3 hydrogen bonds are necessary to pair guanine with cytosine. 2 hydrogen bonds are necessary to pair adenine with thymine. And the diameter is 20 angstroms. Length of DNA is 34 angstroms. In terms of nanometers, 3.4 nanometers. In terms of meters, 3.4 into 10 power minus 9 meters. Now, it is here we have drawn the B type of DNA. B DNA. In case of B DNA, the helix or the coiling is considered as clockwise direction or right-handed helix. Helix is right-handed. It is also used with another term, that is clockwise. Next we will see. Here you have to remember one point, that adenine always pairs with thymine. Means purines are always pairing with thymine. Guanine is pairing with cytosine. Again purine is pairing with pyrimidine. Purine with pyrimidine, purine with pyrimidine. Means here the rule that is followed is Erwin-Chargaff's rule. Is running anti-parallel. Let's write about one type of strand in one direction. DNA is made up of nucleotides. Nucleotides have pentose sugar, phosphate molecule, and nitrogen base. So, first I am drawing pentose sugars. I will give carbon numbering 1, 2, 3, 4 and 5 carbon number 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. See, in case of pentose sugar, that is in DNA, that is deoxyribose pentose sugar, first of all, we will again recall the structure of pentose sugar. Here the structure is OH, H, and here is H, H, and here is H, H. Here it is OH, and here it is CH2OH. In this small DNA fragment, this end of DNA is ending with 3 carbon position, that is hydroxyl group, that is on the 3 carbon end of the pentose sugar. So we will label this end of the DNA as 3' end. This end of DNA fragment means one strand we are writing as 3' here and here. Then what is meant by 3'? For suppose think this is one complete strand of DNA. This DNA small strand is having one end with 3 carbon position hydroxyl group. It is ending there. Nothing is there extended. Such end is called as 3' end of DNA fragment. That only I have written as 3' end. See here in this pentose sugar, which is in the beginning, the end of the pentose sugar here is having CH2OH group containing 5 carbon position. We can write this one as 5' end. 5' end. Now, this strand runs from 3' to 5' for suppose think. We will join these strand links later. Let me give out arrangement of pentose sugars in the side by strand, but we are telling it is anti-parallelly running. Okay. Again pentose sugar. See, I am writing in reverse. Because it is anti-parallel, one end if is going from down to top direction, other strand has to move from top to bottom. The same pentose sugar I have written in upside down direction. We will give carbon numbering carbon 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. Let's join these two strands which are running anti-parallel. See here also, this carbon, fifth carbon position is ending with CH2OH here. So we can write this one as 5' end. Here also, this is ending with OH group. We can write here as 3' end. 3' end. Okay. Now we will join these two strands. This is having OH group here. This is also having CH2OH at the fifth carbon position. But a phosphate molecule has to be present, that is connecting two nucleotides. Okay. Now, phosphate molecule is having one double bond O oxygen and 3 number of hydroxyl groups. So, one OH, other OH, other OH are present in case of phosphate molecule. In the previous biomolecules topic, when we are discussing about this nucleic acids, I have told you the hydrogen of the phosphate is binding with the hydroxyl group of the 3' end of one pentose sugar, and it is being removed in the form of water. Removal of water is called as dehydration. So that oxygen that is present in case of phosphate molecule forms. So when water molecule is removed, it forms an ester bond. It forms an ester bond. Here also, this hydrogen of the phosphate, hydroxyl of the fifth carbon position, this is also being removed as water molecule. And here also is formed another bond, that is ester bond, between oxygen and carbon position at the fifth carbon position. This we will write a little bit clearly. Okay. So this bond is also called as ester bond. So one phosphate molecule to form or to connect two pentose sugars which are one below the other or one above the other with help of 2 number of ester bonds. So we call phosphate molecule is having diester bond in the nucleotide, in the nucleic acid strands. The same I am writing again and again. Oxygen, phosphate, oxygen like this. Now, here also, this is first carbon of the pentose sugar. Suppose it is having nitrogen base, any of those nitrogen base. We know that the bond that is present between nitrogen base and the pentose sugar in a nucleotide is glycosidic bond. Glycosidic bond. Here also, another nitrogen base. If it is adenine and thymine, it will be bonded like this. Guanine, cytosine. Next. Thymine, double bond, adenine, again glycosidic bond. So here. So now, this is about the double-stranded helical form, and this is known as biochemical structure of the double-stranded structure. Again, I am writing, this is biochemical representation. Structure of DNA, double-stranded DNA. And I think you understood what is meant by 3' and 5'. When you know the pentose sugar complete structure and it also biochemically, then you will understand what is this 5' and what is this 3'. Here also see, one end is having 3'. The same there nearby I have written as 5'. This is 3' and this is 5'. Let's see the measurements also. Here we have written this strand is running from 3 prime to 5 prime. Then other strand will run in opposite direction from 5 prime to 3 prime direction. So here we are seeing this strand is running from 3 prime to 5 prime. Then other strand again in the opposite end. So again it is 3 prime to 5 prime but in opposite direction. We have seen here length of one turn is 34 angstroms. Then what is the distance between two nitrogen bases which are side by side like this? So if we write a structure between, for our understanding, between two nitrogen base pairs, this length is considered as 3.4 angstroms. The length between two nucleotides we can also call, or between two nitrogen bases, 3.4 angstroms, or how we have written just before in terms of nanometers, 0.34 nanometers or 0.34 into 10 power minus 9 meters. Next, how we finalized the diameter of a double-stranded DNA. See the distance between two phosphate molecules. Here is the phosphate molecule. We will start from here. And the line that is equal to that of, corresponding to that of phosphate molecule on other strand. The distance between this phosphate and the line corresponding to the phosphate which is nearby to this distance is considered as 20 angstroms, means nothing but diameter. So the diameter is calculated between the distance of two phosphate molecules which are on either side of the DNA, anti-parallelly running double-stranded DNA molecule. We will see some of the points regarding DNA now. DNA, we know that it is having genetic information and that flows in a particular direction. So, DNA has genetic information. And in molecular biology, the transfer of the information from DNA till the formation of proteins. That concept is proposed by Crick. The concept of transfer of genetic information was proposed by Francis Crick. What term did he give about this transferring of the genetic information and the direction of a transfer? So, direction of genetic information transfer. This total sentence meaning is given with one single term, that is central dogma, which also is simplified by writing DNA has genetic information that is coding for RNA, type of RNA, means the information has been transferred from DNA into RNA by a process called as transcription. Now, the information that is present on the RNA, which is taken from DNA, is helpful for synthesis of proteins by a process called as translation. So, the total process from DNA till the formation of proteins is called as, the total process is called as central dogma. Now, there must be some small DNA fragment that is responsible for the total central dogma. So, the DNA or DNA fragment that is responsible for central dogma is termed as cistron. So first priority to apply the term cistron is to DNA fragment. But we also apply this one later to mRNA also when it is giving out the proteins or polypeptide chains, number of polypeptide chains. Basing on that, again the terms changes like monocistronic and polycistronic. Now, if the mRNA that is coded by DNA is coding for only one type of protein chain, means one mRNA coding for one type of polypeptide chain, such a type of cistron is called as monocistronic. If a piece of mRNA or if a mRNA, you write, if a mRNA or, because we can apply this term for both DNA and RNA also. But if mRNA with one promoter sequence, one terminator sequence, coding for one type of polypeptide chain at a time, it is called as monocistronic. Or a DNA fragment that codes for one type or one number polypeptide chain is called as monocistronic. Since this mRNA is coded from DNA only during the process of transcription. Example for monocistronic are eukaryotes. So eukaryotes have monocistronic type of mRNA, and the cistron that is responsible for monocistronic nature is the DNA fragment or the cistron. A mRNA or DNA fragment that codes for many polypeptide chains, then termed as polycistronic. We should remember that either monocistronic or polycistronic type of mRNA has only one promoter sequence and one terminator sequence. Polycistronic in the term means there are not many type of promoter sequences. One promoter sequence plus one terminator sequence. In polycistronic also, one promoter sequence, one terminator sequence. Now, this polycistronic condition is seen in case of prokaryotes. Then how it is possible means, it is having an adjacent side-by-side arranged promoter sequences. One mRNA is having only one promoter sequence. But again, there are many nearby promoter sequences which can come and bind on to mRNA to carry on. But the mRNA is one, but the promoter sequence keep on changing. That type of genes are called as contiguous genes. Here, mRNA with promoter sequences or promoter sequence has contiguous genes. Means once the same mRNA promoter sequence has coded one type of protein chain, again another promoter sequence will be changed. Those genes will be coming and binded there at the promoter sequence region. Contiguous genes. So this is about cistron, monocistronic and polycistronic. It is applied to small chromosome fragment that undergoes mutation. The chromosome unit or fragment that undergoes mutation. Another one, recon. Recon is the term that we apply for the genes that majorly code for enzymatic type of proteins. The gene sequence that code for enzyme form of proteins. I forgot to tell you one more point, that is about cistron. Is it double stranded or single stranded? Cistron is double-stranded in nature. Double-stranded in DNA. C value. It means the total DNA in a set of chromosomes. Here we are using the term set. It doesn't mean that always 2 in number. But suppose gametes, their nature is existing only one chromosome inside them, so we are calling gametes are haploid. Still we have to use a term like one set of chromosomes are present. This is a special type of terminology that we use here while reading about genetics. So here, example, gametes have one set that we represent with small n. Again, zygote you take, which is diploid in condition, so we write 2n. Now, C value is represented with C capital alphabet. So, one set of chromosomes means the C value is 1C only. If diploid cell is having 2 number of chromosomes means, so here the condition is diploid in a cell, then what is the C value means 2C. Now what are units to calculate this C value? Units for C value are picograms. We represent with PG. Picograms. Next. This is about C value. Now, there are some types of human genome, means the total genetic content in human being, that is in a cell, that is considered as human genome has 3 billion number of base pairs. These are some of the basic terms that we have to learn. Next, some DNA fragments, they are mobile in nature. They are called as jumping genes. Next, another term for these jumping genes is also called as transposable genes. Genes or transposable elements that we represent with capital T, capital E, small s. These jumping genes or the transposable elements, they move from one location of the genome to other location. They move from one location in a genome to other location. For suppose, in case of sperms and egg cells, nothing but the gametes, these jumping genes are existed and they jump around the genome within that sperm cell or within the egg cell, and that movement or that transportation or mobility is very much important for the course of evolution. During evolution, total genetic material within a cell. It can be an egg cell or normal somatic cell or anything, but the total genetic material inside that is called as genome. So DNA basically is double-stranded. So normal organisms we can consider exceptions for, but some bacteriophages like pi viruses and also 5X174 viruses which affect E. coli, have single-stranded DNA molecule. In the same way, RNA is basically single-stranded. Single-stranded I am writing with 2S. So SNS, single stranded RNA. So here also there is one exception. So exception is reoviruses. Reovirus that has double-stranded RNA. Double-stranded I have written as small d, small s. So these are few points regarding the DNA. Next, another important term is also there. What is meant by naked DNA? Naked DNA. In case of eukaryotes, the DNA is highly coiled around special type of mechanical strength giving structures called as histone proteins. But in case of prokaryotes, those histone proteins are absent. But DNA is embedded in a gelatinous material-like structure that we call as nucleoid or genopore. So, DNA without histone proteins and not covered by nuclear membrane but embedded in gelatinous material altogether termed as nucleoid. Now, or genopore. The DNA that is present in such nucleoid is called as naked DNA. DNA in such nucleoid is termed as naked DNA.